Hi everyone. Today I will be presenting our work on the semi-deconvolution of bulk and single-cell RNA-seq data with application to metastatic progression in breast cancer. The term cancer metastasis refers to the spread of cancer cells from the primary site to metastatic sites. It is responsible for the majority of deaths by cancer. And throughout this metastatic progress, we can observe a lot of variations in both the cell population composition and the gene expression profile specific to each cell population. Those variations are very informative for cancer progression modeling and understanding the mechanism underlying the cancer metastasis. Traditionally, it is very intuitive to profile those changes in cell populations using single-cell RNA-seq data. So the single-cell RNA-seq data will just be a cell-by-gene matrix, and using a series of cell population biomarkers, we can assign each cell with a cell population label. We can then go on to calculate what is the gene expression profile or the average gene expression for each cell population, or calculates what percentage of cells come from each cell population in the tissue sample. However, this single-cell RNA-seq assay is not very feasible for archived tissue samples. So in a longitudinal analysis of cancer patients, we will usually have the archived tissue samples from the patient's primary tumor site, but the fresh tissue samples from the patient's metastatic sites. In those longitudinal analyses, because single-cell RNA-seq assay cannot be reliably performed on archived tissue samples, we cannot fully rely on single-cell RNA-seq data to profile the cell populations. So to address this issue, this work focused on developing methods to infer similar information regarding cell population profiles from bulk RNA-seq data. Before we jump into how we go about doing that, let's first talk about what is the deconvolution for bulk RNA-seq data. So the bulk RNA-seq data is just a matrix B that is a sample by gene matrix. And we hypothesize that the matrix B is just the product of two other matrices. The first matrix being the expression profile C that describes the unique gene expression profile for each cell population. And the second matrix being the cell type fraction matrix F, describing what percentage of cells come from each cell population in the tissue sample. So the process of multiplying C and F to get the bulk RNA-seq data matrix B is called convolution. So reversing this progress to infer the expression profile matrix C and cell type fraction matrix F from the bulk RNA-seq data matrix B is then just called deconvolution. In our previous work, RAD, we proposed a method to infer C and F matrices from the bulk RNA-seq data matrix B by optimizing the objective function shown here. Essentially, we're trying to infer C and F matrices that will minimize the difference between B and the product of C and F. And in this work, we're trying to extend our work in RAD by adding an additional penalization term to the objective function. And we're calling this method RADS, RAD super. In this setup, we will have the bulk RNA-seq data from the primary tumor site of the cancer patients, as well as the single-cell RNA-seq data from the metastatic sites of the same cancer patients. We acknowledge that there might be some cell types that are only present in the bulk RNA-seq data from the primary tumor site, but not present in the single-cell RNA-seq data in the metastatic sites. So we're representing the gene expression matrix C as a horizontal concatenation between two different matrices, C2 and C1. C2 represents the expression profile of cell types that are missing in single-cell RNA-seq data, and C1 represents the expression profile for cell types that are present in single-cell RNA-seq data. In this penalization turn, we're also trying to minimize the difference between the inferred C1 and the expression profile that we got from the single-cell RNA-seq data. We're also accounting for the magnitude difference in the measured gene expression between the bulk and single-cell RNA-seq experiments using the scalar value mu to scale the S matrix. The penalization effect of this additional term is controlled by the coefficient lambda. In this objective function, only B and S are known matrices, and we will have to infer C, F, and mu. To optimize for the three variables in the objective function, we're dividing the optimization process into three phases. In the first phase, we're only optimizing for the fraction matrix F. And in this subproblem for this phase, we're fixing the C and mu terms. We can then reformulate the subproblem for this phase into one that essentially minimizes the error for each column of B minus the product of C and F. And this problem is then in the same formulation as a quadratic programming problem and can be solved by a quadratic programming solver. In the second phase, we're fixing F and mu to only optimize for this gene expression profile by cell type matrix C. Recall that C2 is just the gene expression profile matrix for cell types that are only present in bulk samples, and C is just the horizontal concatenation of two different matrices, C1 and C2. We can then reformulate the subproblem for this phase into this formulation. We can then solve this optimization subproblem by iteratively optimizing for C1 and C2 matrices using coordinate descent. In the last phase, we're only optimizing for the scaling factor mu. Recall that mu is just a scalar value, and at this point, C1 matrix has already been optimized. S is a known matrix that we can derive from the single-cell RNA-seq data. Then the subproblem for this phase is essentially just a linear regression problem. And we can intuitively optimize for mu by finding a value that minimizes the residual sum of squares between S and C1 matrices. Having developed the RADS method to deconvolute the C and F matrices from bulk RNA-seq data, we can then apply the method on different data sets. The first data set is a simulated one. It consists of the bulk RNA-seq data from the primary tumor site of the cancer patients and the single-cell RNA-seq data from the metastatic site of the same patients. Because this is a simulated data set, we'll have the known ground truth C and F matrices to compare our inferred matrices to. The purpose of this simulated data set is then to evaluate the accuracy of RADS on inferring the C and F matrices, as well as the scalar value mu. Having verified the performance of RADS, we can then apply the method on a real-world data set. In this data set, we'll have the bulk RNA-seq data from the primary tumor site of a breast cancer patient, as well as the single-cell RNA-seq data from the two bone metastatic sites of the same patient. We do not have the known ground truth CNF for the real-world data set, but we can use it to profile the cell population composition and gene expression changes happening in the metastatic process. In the first experiment, we're hoping to evaluate the accuracy of RADS on the simulated data set. In this experiment, we're comparing RADS against a few other methods. The first method being compared to is RAD, which is our previously developed method for deconvoluting bulk RNA-seq data without information from the single-cell RNA-seq. We are also including RADS with the lambda set to 0 for comparison. When lambda is set to 0, RADS is not affected by the penalization term, so it is also not using single-cell RNA-seq as a guidance. We're including both RAD and RADS with a lambda of 0 because their optimization process is different. Additionally, we are also including DSA, which is another deconvolution method that also uses prior information about cell type specific gene expression profile. In this experiment, we're evaluating the performance of RADS by calculating the L1 loss between the inferred C and the ground truth C, as well as the mean square error between the inferred F and the ground truth F. In this experiment, we can see that the RADS with a non-zero lambda is having a lower L1 loss on the C inferred compared to the other methods. It is also having a lower mean square error on the inferred F compared to the other methods. In this data set, the gene expression profile S inferred from the single-cell RNA-seq data deviates from the gene expression profile from the bulk RNA-seq data sets. However, in those cases, the RADS with a non-zero lambda still outperforms the other methods. This is showing that the performance of RADS is unbiased even when S deviates from C. Previous deconvolution methods usually show poor performance when the number of bulk samples is limited. And here we're applying RADS on the simulated data sets that includes only one bulk RNA-seq sample to be deconvoluted. And we're also comparing the performance of RADS with and without S. In this experiment, the L1 loss on the inferred C and the mean square error on the inferred F of the RADS with S are both lower than the results that we got from the RADS without S. This is showing that the guidance from the single-cell RNA-seq sample can be very helpful when the bulk RNA-seq samples are limited. And then we benchmark the performance of RADS against two other state-of-the-art deconvolution algorithms that also take advantage of prior information about cell type specific gene expression profiles. Those methods include CyberSource-X and DSA. On our simulated data sets, RADS is showing a lower mean square error on the inferred F and the L1 loss on the inferred C compared to the other methods. This improvement can be due to the utilization of the single-cell RNA-seq data from the same patient by RADS. Additionally, RADS can also profile the unknown cell populations that are only present in bulk samples. Lastly, we apply RADS on the data set from a single breast cancer patient. This is a real-world data set, and we have the bulk RNA-seq data from the primary tumor of the patient, as well as the single-cell RNA-seq dataset from the 2 bone metastatic sites of this patient. By this experiment, we're hoping to reveal some systematic changes in the breast cancer metastatic microenvironment. In this figure, we're showing the changes in the fraction of cells from different cell types from the primary tumor site to the metastatic sites. The A over M refers to the average cell population fractions in the 2 bone metastatic sites. We could see a larger proportion of fibroblasts present in the bone metastatic sites than the primary tumor sites. This is consistent with previous finding that the cancer-induced fibroblasts can contribute to the cancer growth and cancer metastasis. We could also see an increase of myeloid cells in the metastatic sites compared to the primary tumor sites. A closer examination of the single-cell RNA-seq data from the metastatic sites suggests that the vast majority of those myeloid cells are actually macrophages. This is also consistent with previous finding that the macrophages will increase in the metastatic samples compared to the primary tumor sites. We also conducted a gene ontology pathway enrichment analysis using the top 1% most down or up-regulated genes in the metastatic samples compared to the primary tumor site. We observed a upregulation of CCR1, CCR5, and IgG binding pathways in the metastatic samples compared to the primary tumor site. This is consistent with previous reports that increase in CCR5 activity will lead to increased homing behavior to metastatic sites in breast cancer. Similarly, the knockdown of CCR1 has been experimentally shown to inhibit the metastasis of breast cancer. Additionally, the increased expression of genes enriched for the binding of IgG has been hypothesized to be a driver of the breast tumor metastatic process. In conclusion, we proposed RADS, which is a toolkit that accurately and robustly profiles the gene expression of cell populations matrix C and the fractions of populations matrix F. We have shown in our experiments that the consideration of single-cell RNA-seq data as a reference S improved the deconvolution accuracy when the number of bulk samples is limited. However, our current method still requires the paired samples from the same patient, which is not a common case. In the future, we're hoping to extend the work of RADS by incorporating third-party reference data that other than the same patient one for a better generalization. Lastly, we would like to acknowledge Dr. Xiaoyan Guo for his very significant contribution to this work, as well as our research advisors, Dr. Russell Schwartz, Dr. Adrian Lee, and Dr. Stefan Osterreich. We also want to acknowledge Dr. Yifeng Tao for his contribution to the previous work, RAD, as well as Kai Ding from Dr. Lee's lab and Xuesong Fu for Dr. Schwartz's lab. This is all for the presentation. Thank you so much for listening to our work.